So prokaryotic cells, within the three domains of life, prokaryotic cells are two of those domains. These are generally the smallest and the most simplest of cells. Their DNA is not enclosed in a nucleus, and they have no organelles, so it is an open floor plan where all of the functions occur throughout the cell, and there is no compartmentalization of function. Some examples of some prokaryotic cells: they can be rods, they can be spirals, they can be spheres, they can function in they can perform photosynthesis, and they can live in colonies. Four structures that are found in all prokaryotic cells are the plasma membrane, the cytoplasm, DNA, which is found as circular loops, ribosomes. Some additional structures that are found in prokaryotic cells are the cell wall, also found in plant cells, not animal cells, the glycocalyx, pili, and flagellum. The plasma membrane in both prokaryotic cells and eukaryotic cells defines the cell as a distinct entity. It defines the cell's outer boundary. It governs its interactions with other cells, and it maintains differences in the chemical composition between the extracellular fluid and the intracellular fluid. And it controls the passage of materials into and out of the cell. The plasma membrane is the majority of the plasma membrane, or its main component, is that of a phospholipid bilayer, where the phospholipids are organized with the hydrophilic phosphate heads being exposed to the water, and the hydrophobic tails being sequestered on the inside, where they have limited exposure to the water. This membrane also gives the phospholipid bilayer its fluid-like properties, where one layer can easily float on another layer. The glycocalyx can exist as a capsule or a slime layer. Glycocalyx, glyco meaning carbohydrate, calyx meaning coat, is a covering that surrounds the cell exterior of some bacteria and other types of cells. The capsule is composed of polysaccharides firmly attached to the cell wall. When we say firmly attached, I would assume that this means that it's covalently bound to the cell wall. The slime layer is composed of glycoproteins. So a glyco, remember, carbohydrate, and protein, So this is a carbohydrate protein complex that's loosely attached to the cell wall, possibly through ionic and hydrogen bond interactions. The cytoplasm is a semi-fluid solution composed of water and inorganic and organic solutes encased within a plasma membrane. Since there is no membrane-bound nucleus, the nucleoid within a cell is where the DNA is located within prokaryotic cells. Plasmids are additional extrachromosomal circular rings of DNA. Because of their feasibility in being transferred from one bacteria to another bacteria, they are routinely used in biotechnology as a vector to transfer DNA. General Paisley, stop. Ribosomes are enzymes that are involved in protein synthesis. They have two subunits, and they are found throughout all three domains of life and in both prokaryotic and eukaryotic cells. Thylakoids are small sacs containing chlorophyll and other pigments that absorb solar energy that can be used to synthesize carbohydrates. So thylakoids are small sacs that contain chlorophyll and other pigments that absorb solar energy and then use that energy to synthesize carbohydrates. This is the definition of photosynthesis. So thylakoids are present in some prokaryotic cells, such as cyanobacteria, and in all plants. Some other structures that are observed in prokaryotic cells are flagella, which are appendages that are anchored in the cell membrane and plasma membrane and allow for a motility of prokaryotes. Fimbriae, or common pili, are small bristle-like fibers that sprout from the cell surface. They allow prokaryotic cells to attach to surfaces. Conjugation pili are rigid tubular structures that are used to exchange DNA among prokaryotes. So they use this pili-like structure, little tube-like structure, to transfer sequences of DNA from one bacteria to another bacterium, or from one prokaryotic cell to another prokaryotic cell. Eukaryotic cells, in contrast to prokaryotic cells, have internal membrane-bound organelles, such as a nucleus, where each has specialized functions. Eukaryotes have a compartmentalized floor plan. What this means is that different sections within the cell, or different organelles within the cell, can have specific functions and specialized functions. Within the domain of eukaryotic cells, there are the four kingdoms that we talked about in the first chapter: the plants, animals, protists, and fungi. One thing that is found in eukaryotic cells are what are called pseudopodia, or false feet. Pseudo meaning false, podia meaning feet, so false feet. Some prokaryotes and other types of other eukaryotic cells, including our cells and unicellular organisms like amoebas, form what are called pseudopods. These are temporary, irregular lobular projections that function in locomotion and prey capture. So these are extensions of the cell that can move away from the main body of the cell. And when they move away and continue to crawl forward along the surface, they drag the rest of the cell with them, allowing cells to move along a surface. Pseudopodia, the way they are formed is the polymerization and depolymerization. Remember polymerization, talking about attaching individual subunits to make a polymer. Depolymerize meaning to degrade that polymer. So pseudopodia move as microfilaments within the cell, which are more made of actin subunits, tubulin subunits. Are built and broken down inside the cell, and the combined actions drag the plasma membrane with the rest of the cell along the surface. An example of pseudopodia is these pictures of an amoeba capturing a prey using pseudopodia. It is believed that eukaryotes evolved from prokaryotes in a series of stages. The first stage was that the ancestral eukaryotic cell gained a nucleus by invagination of the plasma membrane. And this invagination of the plasma membrane surrounded the DNA. Allowing this surrounding and compartmentalization of the DNA from other regions of the cell allowed for the nucleus to take on specific functions and freeing up other cellular resources within the cell for other functions. The cell gained an endomembrane system by proliferation of that same membrane that was invaginated into the cell. This endomembrane system, remember how we like to have surface area in a cell. We like to maximize our surface area to volume ratio. This endomembrane system allowed the cell interior to have increased surface area. And increased surface area results in that higher rate of transport or diffusion of materials within a cell. Afterwards, the cells gained, this same cell that had the nucleus gained mitochondria through endosymbiosis of mitochondria, and it gained chloroplasts through endosymbiosis. What this is it would be similar to this ancestral cell with just the nucleus using pseudopodia to take in and internalize an aerobic bacterium like mitochondria. Aerobic meaning it requires oxygen. This is called endosymbiosis because the bacterium are internalized into the eukaryotic cells. The cell, through gaining mitochondria, develops the ability to metabolize sugars in the presence of oxygen, and this enables for greater function and success of that cell. Through taking in the chloroplasts, the cells gain the ability to produce sugar from sunlight and enable greater function and success. So, endosymbiosis is where an ancestral prokaryotic cell, a mitochondria or chloroplasts, that are proficient in converting food and oxygen into energy are engulfed by a eukaryotic cell or a prokaryotic cell that will eventually become a eukaryotic cell. And at that point, the ancestral eukaryotic cell and the prokaryotic cell merge. And over time, the engulfed prokaryotic cell evolves into an organelle such as a mitochondria or a chloroplast. Invagination is where the membrane, the plasma membrane, folds in on itself. And the endomembrane system is developed when there's a proliferation of that membrane within the cell. With endosymbiosis, if you think of the word symbiosis, it means that both parties in an interaction are benefited. What would be the benefit for the mitochondria or the chloroplast in this case to be taken in by this invagination or endosymbiosis? They are protected from harm. Within a bigger cell, they're less likely to get eaten by other things, and they receive shelter. Also, they are a benefit to the rest of the cell, because that cell now has the ability to use oxygen to break down sugars to get energy. And there's a compartmentalization of function. So the nucleus is believed to have evolved by invagination of the plasma membrane. This process also explains the origins of the endoplasmic reticulum and the Golgi complex, which are parts of the endomembrane system. So the endomembrane system includes the nucleus, the endoplasmic reticulum, and the Golgi complex or Golgi apparatus, depending on who's talking about it. Energy organelles, like mitochondria and chloroplasts, may have originated when a eukaryotic cell engulfed smaller prokaryotic cells. The eukaryotic cells would have benefited from this ability to utilize oxygen and or synthesize organic food, endosymbiosis. This idea that chloroplasts and mitochondria were developed into organelles in the form of endosymbiosis is supported by the fact that these organelles, like well-known prokaryotic cells, have their own DNA, their own RNA, and ribosomes. And both of these types of organelles function in creating energy within the cells that they reside. This is a depiction of a representative plant cell. Within both plant and animal cells, which are both eukaryotic cells, there is a nucleus, plasma membrane, ribosomes, mitochondria, rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi apparatus, cytoplasm, cytoskeleton, and lysosomes. Structures that are not found in plant cells, found in plant cells but not in animal cells, are chloroplasts. The cell wall, remember that's something found in all prokaryotic cells. And vacuoles that are occasionally found in animal cells, but not this large of a vacuole as the one observed in this plant cell. Sorry. Within a representative animal cell, we have chromatin, we have a nucleolus, which is where the ribosomal RNA, rRNA, and ribosomal proteins are synthesized. A smooth endoplasmic reticulum, cytoplasm, mitochondria, lysosomes, centrioles, which we do not have in plant cells, centrosome matrix. We have vesicles that are secreted using exocytosis, which means to have something exit the cell. Golgi apparatus, ribosomes, rough endoplasmic reticulum. The eukaryotic endomembrane system compartmentalizes certain functions within this eukaryotic cell. It is composed of different membranes that enclose the nucleus, the smooth endoplasmic reticulum, and the rough endoplasmic reticulum, and the Golgi apparatus, and other vesicles that are suspended in the cytoplasm. In conjunction with the cytoskeleton, this system is involved in cellular transport of substances. Eukaryotic cellular organelles. The first one, the nucleus, is the largest organelle. It contains the genetic library of blueprints, aka DNA, for the synthesis of nearly all cellular proteins. It responds to signals dictating the synthesis of the different kinds and amounts of proteins. Most cells are uninucleate, meaning they have one nucleus. Some cells are multinucleate, meaning they have multiple nuclei. And there's one type of cell that I know of right now that is anucleate, which is the red blood cell. Composed of the nucleus, there are three structures: the nuclear envelope, the nucleoli, which remember that's where the ribosomal RNA is created in the ribosomal proteins, and chromatin. Chromatin is the cell's collection of DNA and associated proteins. A chromosome is one chromatin molecule and all of its associated proteins. The chromatin structure changes as the cell divides. The nuclear envelope, or membrane, is two continuous lipid bilayers folded on each other. Continuous, this is different from the plasma membrane. In the plasma membrane, the two layers are two separate layers. They are not folded, so it's one continuous layer. The innermost surface of the nuclear envelope has DNA attachment sites, and the spores span the bilayer. So one layer of this continuous layer of lipids one layer faces the cytoplasm, and one layer with DNA attachment sites faces the nucleoplasm, which is similar to the cytoplasm but is within the nucleus. Nuclear pores transport molecules across the nuclear envelope. They transport RNA and ribosomal proteins, proteins such as DNA polymerase, an enzyme which we'll talk about in the next week, carbohydrates, signaling molecules, and lipids. The nuclear envelope, or the nuclear lamina, is, remember we talked about the DNA, just in a previous slide we talked about the inner layer of the nuclear envelope having DNA attachment sites. Within that region of the DNA attachment sites, there is this nuclear lamina, which is this dense fibrillar network inside the nucleus of most cells. It is composed of intermediate filaments, membrane-associated proteins, and it functions in providing mechanical support to the cell and regulating important events such as DNA replication and cell division. So it lies on the inner surface of the nuclear envelope. The nucleolus is this dense mass of material in the nucleus, and it is a cluster of DNA and proteins. The nucleus may have more than one of these nucleoli, where this is where the ribosomal RNA is transcribed and assembled into ribosomal subunits. These subunits must pass through the nuclear pores in order to reach the cytoplasm, where protein synthesis occurs. If they function in the cytoplasm, the proteins that they synthesize will stay and function in the cytoplasm. If the proteins are being synthesized and their final destination and their final pathway will involve being secreted from the cell, they are the polypeptides that will enter into the rough endoplasmic reticulum. The endoplasmic reticulum. Within animal cells, the endoplasmic reticulum is continuous with the nuclear membrane, and it extends throughout the cytoplasm. There are two regions called the rough and the smooth cytoplasm. The rough endoplasmic reticulum is arranged into flattened sacs and has ribosomes that riddle the surface of it, giving it this rough appearance. Remember, these are are ribosomes that are functioning in synthesizing proteins that will then enter into the rough endoplasmic reticulum. Cells that specialize in secreting proteins have a lot of rough endoplasmic reticulum. The smooth ER are a series of interconnected tubules. They have no ribosomes on their surface, and lipids are assembled inside the tubules. The smooth ER in the liver also inactivates and detoxifies waste and drugs. There is a specialized smooth ER in muscle that regulates calcium ion concentration in the cytoplasm of striated muscle cells. So we've now talked about the organelles that are involved in the endomembrane system, which includes the nucleus, the smooth and the rough endoplasmic reticulum, and the Golgi apparatus. Vesicles are membranous sacs that move material throughout the cytoplasm. We have like vesicles that are pinched off from the rough endoplasmic reticulum that will then fuse with the Golgi apparatus, and vesicles that will be pinched off from the Golgi apparatus. So we have some specialized vesicles, like lysosomes that contain digestive enzymes. They act as floating garbage disposals for cells. Within these lysosomes, they have a lower pH, so it's a more acidic environment, and they function in digesting and recycling cellular waste products and consumed material. A functional relationship between ribosomes, rough ER, Golgi complex, and secretory vesicles in the synthesis and secretion of proteins. So proteins containing vesicles pinch off the rough endoplasmic reticulum and migrate to fuse with membranes of the Golgi apparatus. Proteins are then modified within Golgi compartments. Proteins can then be packaged within different vesicle types depending on their ultimate destinations. These vesicles can either contain contents that are destined for exocytosis or exocytosis or secretion outside of the cell. They can be vesicles that contain proteins that will become incorporated into the plasma membrane. Or there is a pathway where these vesicles then fuse with the lysosome, which contains these acid hydrolase enzymes that then will break down the proteins. For example, if the protein was misfolded and is not functional, it can be fused with the lysosome, which will then break it down, and the components of that misfolded protein can be used for other protein synthesis. So peroxisomes, membrane-bound organelles that contain oxygen, similar to lysosomes in that but peroxisomes break down fatty acids and some other organic molecules. Lysosomes are more specific for proteins. Vacuoles, like vesicles, they are membranous sacs, but they are larger. They are found in all eukaryotic cells. Protist cells have quite specialized vacuoles that are contractile vacuoles that can rid the cells of excess water. There are also digestive vacuoles for breaking down nutrients. Animal cells have very few vacuoles, but plant cells are lipid-engorged vacuoles. The central vacuole, which is found in plant cells, stores nutrients, retains and degrades waste products, accumulates poisonous products, and contains pigments that enable plants to attract birds and insects that help these plants to reproduce. It can also provide physical support to the plant. Chloroplasts function as the site of photosynthesis, or the conversion of light energy into chemical energy. Chloroplasts are composed of two outer membranes that surround the stroma that bathe an inner membrane. So two outer membranes that surround the stroma and bathe an inner membrane that is folded into thylakoid stacks. Each stack of thylakoids is called a granum. The granums contain chlorophylls and other substances that are involved in photosynthesis. Mitochondria are ATP-producing powerhouses. They have a double membrane system and perform the most efficient energy-releasing reactions. These reactions require oxygen, meaning they are aerobic organelles that require oxygen to perform their actions. The mitochondrial structure is like that of the chloroplast, is a double membrane system where the outer membrane faces the cytoplasm and the inner membrane folds back on itself into invaginations called cristae. These membranes allow for two different compartments to be distinct within the organelle. The Krebs cycle is located in the matrix that's found within the inner membrane. And the ATP generating machinery is embedded in the inner membrane. In the membrane of the inner membrane. The cytoskeleton of a cell is the cell's structural framework. It performs similar functions in a way to our bony skeleton, in that it allows for shape and function. It allows for internal organization. So it is the basis for cell shape, and it allows for organelle and vesicle movement within the cells. It also allows for motility in some cases. For example, pseudopodia are made from microtubules. Their cytoskeleton is made up of actin filaments or microfilaments, intermediate filaments, and microtubules. Actin filaments or microfilaments are the thinnest cytoskeletal proteins. They are made of semi-flexible strands of actin. There is, on the interior of the plasma membrane, there is a dense cross-linked network of microfilaments that are attached to the plasma membrane. It serves to strengthen the cell surface and helps resist compression. It may also be involved in cell motility and changes in cell shape. For example, pseudopodia. It also is involved in allowing materials to enter and exit the cell. When materials enter the cell, they do it through endocytosis, meaning to enter a cell, endo meaning to enter, to come in, cytosis meaning cell, so enter the cell. Exocytosis, exo meaning to exit, cytosis meaning to cell, so this is to exit the cell. Intermediate filaments are filaments that are intermediate in size between the microfilaments and the microtubules. They form tough, insoluble, rope-like strong structures. They are composed of a tetramer of 4 fibril polypeptide strands twisted together. And they serve to provide support to a cell and help cells to resist pulling forces. These filaments attach to desmosome plaques that serve similar to Velcro, that help cells to resist pulling forces when pressure is exerted on their surfaces. So they help form an internal tension-reducing network of fibers. In nerve cells, intermediate filaments are called neurofilaments, and in skin cells they are called keratin filaments. Microtubules are the largest of the cytoskeletal elements. They are composed of hollow tubes made of tubulin. So their individual subunits are tubulin subunits. These tubes are constantly assembled and disassembled, and most radiate out from a centrosome which contains centrioles. They function in helping determine overall shape of the cells, keeping organelles in place by tethering them, and allowing for substances to move within the cell using motor proteins that use the microtubules as tracks to ride along. So these are hollow tubes made of tubulins that are continually disassembled and reassembled. And motor proteins can use them to move substances within the cell using the microtubules as tracks. There's a brief overview of these three different types of components. Microfilaments, strands of spherical subunits called actin. Intermediate filaments that are tough, insoluble protein fibers that are constructed like woven ropes composed of tetramer fibrils. And hollow tubes of spherical protein subunits called tubulin or microtubules. Microvilli, flagella, and cilia. Many cells have surface extensions called microvilli, flagella, or cilia that aid in absorption, movement, or sensory processes. Here's the microvilli. They're an extension of the plasma membrane. Flagella that help in motility of cells, and cilia which help in some motility across a surface of a cell and in sensory processes. So microvilli are extensions of the plasma membrane that serve to increase the surface area of the cell. They are anchored by the cytoskeletal elements, the actin filaments. They also do not contain axonemes. What are axonemes? That's on the next slide. Microtubules in flagella and cilia are arranged in an axoneme, which is a 9+2 microtubule array. What this is that two central microtubules where each consists of 13 protofilaments that are arranged to form a circular tube. Each of the 9 outer doublets of microtubules are two microfilaments combined together that share a common wall. Flagellum are whip-like structures that allow for locomotion of a cell. They are found in all 3 domains of the living world, including bacteria, archaea, and eukaryota. Helicobacter pylori if you've ever know someone who's dealt with stomach ulcers or dealt with them personally, this is often your culprit. This sperm. The hair-like appearance of cilia and flagella is misleading because the entire structure of the flagella lies within the plasma membrane of the cell. And so it's that axoneme, this 2 plus 9, where the 9 are doublets of 2 microtubules that share a common wall, and each of the microtubules is composed of 13 protofilaments. As we move down the flagella, the structure of the flagella changes. So just like this, the end of the flagellum looks like a section of a centriole, or has the same structure as a centriole. When 2 centrioles are found next to each other within a cell, they are found at right angles. For example, here, if we wanted to make a cross section through here, one of them, they are found perpendicular to each other. Centrioles work in pairs, with each moving toward the poles of the nucleus when it is time for cell division. Cilia move and promote the movement of substances across their surface through an alternation of a power stroke and a recovery stroke. So they have this power propulsive stroke, and then there's a recovering stroke where it flips back. And this allows for a traveling wave to be created across the surface of the cells. An overview of the organelles here. So we have, in both animal and plant cells, we have a nucleus which directs the cellular activity and stores hereditary information. The cytoskeleton, which provides structural shape and support and enables cellular movement. Mitochondria, which harvests energy for cellular functions. Lysosomes, which digest and recycle cellular waste products and consume material. Rough endoplasmic reticulum, which modifies proteins that will be shipped elsewhere. Smooth ER, that synthesizes and detoxifies molecules. Golgi apparatus, which processes and packages proteins, lipids, and other molecules. A cell wall, which is found only in plant cells and also prokaryotic cells, that provides structural strength, protection, and increased resistance to water loss. Vacuoles, that are sometimes found in animal cells, but always found in plant cells, where they store nutrients, degrade waste products, provide pigments and structural support to the plant. And chloroplasts, which are found in plant cells but not in animal cells. Remember, they're also found in cyanobacteria and prokaryotic cells. last of this, I want to do a brief review of microscopy. Compound light microscopes use a set of two lenses to focus light rays that pass through specimens to produce an image. So one set of lenses, light is shown through one set of lenses, focuses a beam. The second set of lenses multiplies that, compounds the strength of the first set of lenses in focusing that same beam of light. And the refraction pattern after the refraction pattern is what is observed by the eye, the refraction pattern of the light. A transmission electron microscope has, instead of having a set of glass lenses, uses two electromagnetic lenses to focus electrons passing through a specimen to produce an image on a fluorescent screen or photographic film. Oh boy, I know. The image that's produced, similar to focusing a beam of electrons, the beam is focused by one set of lenses, and then it's sent through another set of lenses that focuses it even further. And then the electrons pass through the specimen, are transmitted or are transmitted through the specimen. And the diffraction, or as the electrons diffract through the specimen and are bent, and their trajectories are bent as they move through the specimen, their trajectories are absorbed by a fluorescent screen or photographic film. That absorption pattern is used to generate the image that's observed. The scanning electron microscope uses a similar set of electromagnetic lenses to focus an electron beam of electrons. But one other thing that's added in this case is that the specimen is coated with a thin layer of electrons. If you remember, we talked about oxidation and reduction, and that electrons can be removed from an atom. The narrow beam of electrons that flows through an electron scanning microscope is aimed at the specimen, and the electrons that are shot at the specimen excite electrons within the metal that are given off by the metal that's coating the specimen. And those secondary electrons that are emitted by the metal are used to create a three-dimensional image of the specimen. So, transmission you use the refraction pattern of electrons moving through the sample. Scanning. You're scanning the surface of the specimen, and with that scanning, you're exciting these secondary electrons that are given off by the metal. And when they bounce off the metal, there's detectors that use that bouncing off pattern of those secondary electrons to create a three-dimensional image of the specimen.